There have been few topics left over in our syllabus. For this purpose, I am sending you this video. Let us talk about protein modeling today. Okay. So why a protein three-dimensional structure is modeled in bioinformatics? In spite of presence of techniques such as X-ray crystallography and NMR, these are the techniques we use to determine the three-dimensional structure of the protein. And in spite of these methods, why does a protein modeling approach why does this approach being used in bioinformatics? Let us understand that. Why is this approach used? The protein structure determination is a quite important aspect to understand the biomolecular function. There have been, which is associated with a lot of difficulties in case of X-ray crystallography and as well as NMR. So in case of NMR, the problem is that the protein is in solution form. And obviously, when you determine the structure, the molecular weight should be less than 40 kilodaltons. A lot of other effects and other defects are there. In case of X-ray crystallography, the crystallization of the protein itself is a difficulty. So because of this purpose, we need to come up with a method where you can predict the three-dimensional structure of the protein without doing any sort of experimentation. That's where the concept of protein modeling has evolved. The protein modeling concept is based on the sequence similarity, which we studied in the sequence analysis part. And this reflects the structural relationships and the sequence information of the protein. Well, to be very precise about what we are going to discuss today is to understand a concept of why these protein three-dimensional structures are being modeled in bioinformatics. What is the need? Now, if you observe that, the major theme or the concept of the protein modeling comes from a protein that scientists have tried to analyze for many of the days. That is about the protein this protein you could have studied in biochemistry. It is called as triose phosphate isomerase. It is involved in conversion of glycerolide 3-phosphate to dihydroxyacetone phosphate and vice versa. It's there in glycolysis. It's there in some steps of photosynthesis as well. You have studied your biochemistry. When you observe the molecular structure of this particular enzyme in different species, such as E. coli, Bacillus stereothermophilus, Plasmodium falciparum, trypanosoma, and yeast, chicken, humans, and all these things, the sequences of this protein differ by 50%. But yet, if you see the structures, they look almost alike, the same. Like you can see in this screen, all the structures from different species of this particular protein are similar. What you're seeing here is a carbon alpha backbone of the protein. That is only the alpha carbons of the protein that have been represented. So based on this, scientists have proposed that if there is certain amount of sequence homology or similarity in the sequence of amino acids, there will be a similarity in the structure among the proteins. So that's what is being used as a step in case of protein modeling. So protein modeling is of different types. We are going to discuss something called as protein homology modeling for your case scenario. Okay. So let us understand how a protein modeling is done. These are the steps that are involved. The first step is identification of structural homologs. Second step includes the selection of structural homologs that are used as templates for modeling the three-dimensional structure of the unknown target. Alignment of these two sequences, then the proposition of the three-dimensional rough model, refinement of the model, that is through loop modeling and sidechain modeling, evaluation and refinement of the model. So all these things are iteratively done until a three-dimensional structure is established. This is one flow chart that shows you how you have to start. So the first step, initial step of any protein modeling technique is like this. You will have a target sequence whose structure is not known. For instance, in this case, you can see a target here. I'll try to highlight this using a pen. So this is the target sequence that I'm having a look at here. And this is the template sequence, which has a 3D structure. You can see here that this has a 3D structure. Now, based on the 3D structure sequence, say the template sequence, which has the 3D structure, and the target sequence homology, we are going to predict the 3D structure for the target sequence. So the first step in protein modeling, as I told you, is to select the template. Template means another sequence which has homology of more than or greater than or equal to 40% with an existing target protein. So this is the one mandate rule that people follow in protein homology modeling. That is, you should have 40% of the similarity among things. Okay. So that's how it is used. So it is 40%, as I told you. Okay. Now, the next step would be the alignment of the sequences between the target and the template, because you should know where the homology is. Okay. So for that, what people do is they use different searching methods like BLAST. In BLAST, there is an option that you can choose a database. Instead of the non-redundant database, you can choose only PDB. So in that case, when you use a protein sequence and search, what happens is that these sequences which are similar to the structures in PDB will be obtained. So that means for your target sequence or unknown sequence, you can get the three-dimensional structure or the template. So this is the template in our case. What you are seeing is 6kpd, and these are target sequence, what you are seeing here. Now, the second step of this protein modeling is to align the target sequence with the template sequence. This is how we'll do. I hope you all know what is sequence alignment. We have learned in the last classes. So based on this alignment, what we understand is that there will be two types of regions that can be obtained. One type of region, we call them as structurally conserved regions. In that case, the amino acid sequence of these regions is quite similar. Like you can see certain regions where the amino acid sequence is exactly precisely the same. The regions which are where the amino acids are the same. For instance, I can mark you a region here. You can see that these two amino acids are same. So these type of regions between the template and target are called as structurally conserved regions, SCR. Okay. There are certain regions. You can see one more structurally conserved region is here, that you can see in this, where this EQ amino acid, glutamine, and the other amino acid is conserved here. So there are certain stretches like that. These are called as structurally conserved regions. On par with that, there are few other regions which are structurally variable regions. We call them as structurally variable regions. Okay. So these two are considered in case of homology modeling. Now, what is the benefit of structurally conserved regions? I'll tell you. Like, let us assume that, there are three different shapes. Like, for example, there is a triangle and there is a square that are inside a circle. So the space that this triangle and the square occupy in one circle of a particular radius and another circle of the same radius would be the same. Do you agree? It will be the same triangle size, same rectangle size, and the same circle radius would be the same in how many ever replicated number of cycles you will get. It will be the same. Now, what we are trying to understand here is when there are two similar amino acids coming up, their XYZ coordinates would be same. Like why? A three-dimensional structure of any protein is represented in form of its XYZ coordinates. So X coordinate, the Y, and the Z coordinate. All these three in a three-dimensional space will tell me where a particular amino acid residue is present in the three-dimensional space. Okay. Now, the same thing that in this case, if you see this L and N amino acid, that is leucine and arginine here, what you have, see this leucine and arginine in case of the target doesn't have a coordinates. It doesn't have a specific coordinates properly folded in a three-dimensional space. But this amino acids L and N in the template has a very good XYZ coordinate combination. XYZ coordinate combination which could be used. Now the point here, what we are looking at in this case, what we are trying to prove around is like this. We are trying to assess that if two amino acids have the same sequence in a stretch, that means their three-dimensional space they occupy would also be the same. That is, their fold would be the same. That's how we can call in the protein language. So based on that, the target three-dimensional model is modeled based on the template. Because you remember, we have given a clause saying that it should have greater than or equal to at least 40 or 50% of homology should be there between the target template sequence and the target sequence. That's what is our mandate is. Okay. Now, the next step would be the model building. In case of model building for the target, because I know the sequence, okay, I'll assume the linear chain of amino acids like this. Can I do that or not? I can. Because, for example, if it is a glycine, I know how the bonding happens between two different amino acids, right? CONH, how it is formed. The peptide bond I can assume. Okay. So I can assume a three-dimensional linear chain of amino acids, which doesn't have XYZ coordinates, some random XYZ coordinates. Okay. I'll assume. Now, what I will try to do is I will assign the three-dimensional coordinates of the three-dimensional template structure to this target structure in the regions where they are structurally conserved. We call that's why it is called as structurally conserved region. That's very important. So we will assign the 3D coordinates of this particular three-dimensional structure to this structure at different structurally conserved patches. Now, because of that, what happens? You can see here, like this is the linear chain of amino acids, the carbon alpha backbone, what you're seeing here. This has been assumed with the three-dimensional coordinates from the template, what you are seeing in this case. So a model building happens, and this results in a rough model. Now, because this rough model is not fully modeled now, it's still partial. Why? Because structural variable regions doesn't have a proper conformation. So for that, we will modify those structurally conserved regions, and we call that intermittent modification or refinement process as loop modeling. Loop modeling is a process that is used to assign a different structure to the structurally variable regions. How it can be done? So it can be done from different types of other components, such as loop databases. We have certain set of databases where fragments of sequences are there, whose three-dimensional coordinate structures are also there. Like we assign based on whatever the variable region is to this variable region, which couldn't be modeled, we will assign a three-dimensional form. So ultimately, because of that, the unmodeled structure becomes properly three-dimensional model. But there is an issue in this case. When you do in loop modeling, in spite of that, there is one more issue. The issue is like this. I am modeling the target from a different template, and the intermittent variable regions I am loop modeling from a different set of structures. So it is like you are trying to use an analogy where you are trying to fit a 10 different pen cap into a different pen. So they will not match up with each other even if I do. For instance, I imagine I have an ink pen, imagine I have a ball pen, I'm trying to use the cap of ink pen to put to a ball pen. It is not properly fitting. This is what I've done till loop modeling is. Now, so what I'm trying to do in this case is that I will do one more function to understand whether my loop modeling has been pakka or not. So I just shown you a small region of patch here, how I have loop model. The initial structure was like this, what you are seeing down here, and it has been modeled into the yellow colored fragment that you are seeing here. That's how the loop modeling works. Now, there are different softwares that are used for loop modeling that are available all over the internet. Okay. So this, do you remember this? We have done in the practicals for you, called as Fiery server, which was used to predict the three-dimensional structure of the protein. Now, how do you evaluate your model, whether it is perfect or not? So the model evaluation is usually done by using Ramachandran plot. You could have heard about Ramachandran plot. Ramachandran plot is a plot of psi-phi angles. What is a psi angle and a phi angle? If you look into a peptide carefully, the carbon alpha backbones are only flexible. Just as the CONH bond, that is the amide bond or the peptide bond that you are looking at, is very rigid and it is partially double bonded in nature. So in that case, the carbon alphas between these alpha carbons between these peptide bonds are very flexible. So because of this flexibility, they can give the bend to the peptide structure. That's what you are seeing, this peptide bend. And that's why these angles are called as torsion angles. What are these torsion angles? There are two torsion angles. One torsion angle, as you see in the zoomed structure now, is between C C alpha and N, which is called as pi. The angle of rotation between C alpha and C is called as psi angles. Okay. So these are the torsion angles, are the angles that give the bend property to the protein. So what Ramachandran and colleague have done is they have plotted the sci-fi angles here. You can see they have started plotting the sci-fi angles here. So how did they plot? They started the plotting of sci-fi angles like this. They started for psi from minus 180 degrees to plus 180, minus 180 to plus 180 for pi. So they plotted psi versus pi combinations and they obtained a plot. This they did it for a simple polyalanine peptide. That is only a peptide, small peptide molecule with only alanines, and they changed the different psi-phase. And what they noticed was that at certain regions, the protein structure was very stable. Like you are seeing, these regions which are colored are very stable. In certain regions where you can see these marked red residues outside the plot, the psi-phase were very bad. Like, these psi-phi combinations have broken the peptide. So they determined which psi-phi combinations are allowed in a protein. So that's what we are going to evaluate in case of a Ramachandran plot. So they evaluated the Ramachandran plot for the protein, and based on this, for all the residues of your peptide, you will calculate the psi-phi angles and you will plot a Ramachandran plot for your entire protein. Now you can see a bad structure in this case towards the left, a refined structure towards the right, because you can see in the bad structure there are a lot of amino acids which are in red color, which are outside, not the region with the red dots, but you can see the names of amino acids which are outside in the white region of the Ramachandran plot. Okay, so that's what is Ramachandran plot, and based on this, scientists will come up with a conclusion that whether the model 3D structure is good or whether it is of low quality. Post this model evaluation, now how do you refine the structure? Because already we have patched two different structures. We have seen certain amino acids are bad. So what people do is they come up with a concept called as energy minimization. What is energy minimization? Let us understand in simple thermodynamic concepts. Like you have three states of molecule, that is solid, liquid, and gas. In solid, the molecules are tightly packed. So the free energy between the molecules is Z, almost very minimum, and it's called as smallest entropy. And if you come to the liquids, they are a little partially packed. So the free energy between the molecules is a little higher than solids, and hence the molecules can freely move and they attain the shape of the container. Their entropy is lower, but the smallest is with solids because these are tightly packed. If you come to the gases, gases have high energy and high entropy. These molecules can't sit in a container. If you open the container of the gas, gas will escape out because of this free energy concept. So they are having high entropy or disorderness. So if a protein is properly modeled, its entropy will be less. That is, its energy will be minimum. So we do a minimization process called as energy minimization. So we use a lot of molecular mechanics, which I'm not going into details. It's not there in the syllabus as well. This is to make you understand what exactly is protein modeling. So we do a process called as molecular energy minimization and molecular dynamic simulations. Both of these things we will use to attain a proper three-dimensional structure of the protein. So this energy minimization is done by using different ways, by setting the calculating the potential energy of the protein, and it's after its structure is set up as per the IUPAC guidelines, how does the potential energy come down? This process is called as energy minimization. They do this energy minimization and the Ramachandran plot evaluation iteratively, even the loop modeling iteratively until they get a proper three-dimensional structure. Analogous to this is one more concept called as molecular dynamics, where the modeled proteins are subject. This is the entire flow diagram of what exactly is what you have done right now. I'll come back to that again. So molecular dynamics is a concept where you simulate the three-dimensional environment for the protein so that it can settle down at a particular good three-dimensional conformation. So anyway, so you have to understand that protein modeling is a process where you produce the three-dimensional structure of a protein through the bioinformatics approach. This saves a lot of experimentation process. So what we have done, let me give you a recap again. You have a target sequence. For the target sequence based on the homology, you found the templates through blast search or different other searches. Then you try to model the target protein structure based on the template protein. After you modeled, then you try to validate the structure using Ramachandran plot. So post-validation, in between anyway, you have done loop modeling, which is a part of modeling as well. And then you came back again for the refinement through energy minimization. These steps are iteratively done. The validation, refinement are iteratively done until a proper three-dimensional model structure is obtained. So this concept is called as protein modeling. I hope you all understood what exactly is protein modeling. If you have any doubts, you can clarify with me anytime.